Let's start with this morning's keynote presentation. It is my pleasure to invite Jeannine Cavender-Bares to give the keynote lecture this morning. She's a Distinguished McKnight Professor at the University of Minnesota and Director of the US NSF Biology Integration Institute Ascend, which seeks to understand the causes and consequences of plant biodiversity across scales using spectral biology. Her work focuses on plant evolutionary ecology, ecosystem function, and advancing remote sensing of biodiversity. She is on the governing board of the Ecological Society of America and a member of the American Academy of Arts and Sciences. Jeannine received her Ph.D. from Harvard University and has studied in Latin America and Europe. She's committed to advancing international efforts for biodiversity monitoring towards sustainability. Jeannine Cavender-Bares, the floor is all yours.
Bonjour à tous et merci beaucoup pour l'invitation de faire la présentation. Je suis très contente d'être ici à Montréal. And I will begin by talking about the interesting times that we are in right now. We're in a moment where we have seen increasing wealth accumulation for humans for hundreds and hundreds of years now. So we are getting wealthier and wealthier. And that's had really important benefits for human well-being. Our life expectancy has increased. We're living much longer than our ancestors did. And that, unfortunately, coincides with the decline of the biodiversity around us, as we all know, as it's the reason we're all here. So as we have been taking more and more from the planet, other species have been getting less and less benefit from the Earth, as witnessed by the very small fraction of total biomass in mammals that are actually wild species. And we've seen from the many best assessments now that biodiversity is declining on every continent. And we've known this. We've known that this was where we were headed, and Chief Seattle told us this a long time ago. So it's really wonderful that this great success in December has led to the Global Biodiversity Framework, and here I've just pulled out 4 of these targets from the first goal that remote sensing can contribute to. And so today I'm going to be talking about the role of Earth observations and how they can help us towards monitoring towards a better future. And so these are the three points that I'm going to talk about today. The first thing is the importance of integrating remote sensing with everything else we're already trying to understand. And I'm coming from a biology background, so in particular I'm interested in how do we integrate Earth observations with really good biological understanding of what's going on. I think the understanding underneath the signals we get from space are really critical to effective monitoring and management of our living planet. The second point I'll make is that the tree of life, which is often seen as a complicated thing that only systematists understand, it's actually a very natural organizing framework for both monitoring and managing life on our planet. And finally, the last point I'll make is that all of this advancing technology and capability without ethics is not going to solve our problems. We really do have to integrate all that technology with an ethic of giving back to our planet, our living planet that's in crisis. So this slide, which Andy Gonzalez kindly gave me, points to the importance of monitoring in advancing policy and conservation. And this has long been the case that we cannot do very well at advancing policy if we don't know what's going on and how things are changing. And when we make policies, what impact does that have? So we need to know the current state of things, how they're changing, and when we make policy changes, what impact that has. So the monitoring part of this management process is indeed really critical. And this is, turns out to be pretty hard to do. It's a lot harder for biodiversity than it is for climate change, for example. And this is just experience I had working on the America's regional assessment for IPBES. I was one of the leads on looking at the status and trends of biodiversity and ecosystem services across all of the Americas, from Canada down to Tierra del Fuego. And we were asked to document how things were changing through time, what the status was and what the trends were through time for all these different biomes or units of analysis. And when we tried to pull the data together, of course, Canada and the US have really good databases. Some countries in South America have really good databases. They're all in different formats. It's really hard to pull the data together. It's really hard to get access to the data if you're not in the right position for that particular country. And there's a lot of gaps. And so we were actually unable to pull together a consistent data set on the status and trends of major biodiversity variables across all the subregions and units of analyses. So right there is a good starting place. We need consistent ways to monitor biodiversity and ecosystem functions and services consistently across the globe without all of these kinds of inconsistencies that make it so hard to do this. And then we have many, many data gaps, as Andy pointed out yesterday. This is a slide made by Kyla Dolan that shows where biologists have measured the functions of plants around the globe and where the hotspots of biodiversity are based on from Conservation International. And those two themselves are not aligned. And also you see the black dots are where most of the biologists actually are, where all the cities are. We're not getting a lot of observations in places where most of the biodiversity is, and this remains a challenge for us. Luckily, we've had major advances in technology in the biological sciences. We've seen the genome revolution, where we can sequence at low cost, really understanding the genomic fundamentals of life on Earth. And at the same time, we've also been able to assemble the tree of life in a way we never could before. We know how species are related to each other. We know who's descended from whom. And I'll get back to this later, but this creates a very important organizing framework for understanding life. And then we've had all these major advances in satellite technology, sensors, and remote sensing capability, not just in space, but also on the ground, many different platforms. And so we're seeing soon launch of new missions like the hyperspectral SBG, Surface Biology and Geology mission, and CHIME, many other satellite missions that are going to give us new capabilities of monitoring life on Earth, especially when coupled with observations at other scales and as we continue to do the really important work on the ground. And so this is the vision that we've been seeing for the global biodiversity observation system. And it's really going to require a lot of work from everybody around the globe. staying focused on what we actually understand and can measure from the ground and coupling that with observations from space and other platforms. And here's just one example of why integration is so important. This is just a table that I've adapted from a paper that you can look up that we published last year, just showing that for every target, particularly this is goals A and B in particular, remote sensing is actually pretty critical. You can't do a lot of this without remote sensing, at least not comprehensively, but it's not sufficient. We absolutely need in situ data. Often we're needing socioeconomic data, often we're needing laboratory data, and all kinds of other information. And so remote sensing alone is not going to solve our problems. We have to do the careful work of integrating, and that's actually hard. And so I've been leading this Biology Integration Institute funded by the US National Science Foundation called Ascend that you heard about before. And so this is a group of people. It's a combination of biologists and remote sensing experts of various kinds. And we're trying to tackle this integration problem in the biological realm, spanning biological scales from genomes to whole plants to species across the tree of life, all the way up to the biosphere. And we're using spectral tools across a range of platforms. But the spectral information gives us a common data type to work across scales. And so in doing that work, we're doing a lot of work in the field and with different platforms and in the lab, and all of these approaches to try to get an understanding of the biological processes that underlie the signals that we get from remote sensing. And here's one example, using neon data from airborne hyperspectral data across the neon sites, neon domains. And in this approach, my colleague Jesus Pinto-Ledesma is able to use the spectral species concept, taking spectral information from all of these sites and use that spectral species information to predict other measures of diversity using a Bayesian regression approach. And it does well in monitoring in predicting species richness, phylogenetic richness, functional richness, and also evenness, and also other metrics of diversity that use higher Hill numbers. And then my colleagues at the University of Wisconsin have really pioneered approaches for mapping functional traits at large spatial extents, using spectral information that is really good at predicting a suite of different functional traits, and then mapping those using the neon data across the United States. But here's the point. This is really hard to do well. Really understanding what's going on biologically is hard to interpret from signals. We can't just take those signals and know what's happening, because ecosystems differ, and the drivers of change and the driver of the various ecosystem processes in different ecosystems differ, even when they're in the same biome. So here's Minnesota and Nebraska, same grassland biome, but the soils are really different. In Minnesota, we've got these very sandy soils, and in Nebraska, these loamy, rich soils with much higher productivity. So in the Minnesota system, we've got biodiversity experiments in each case where we can try to tease apart the factors that drive below ground processes. And here we're trying to actually understand what is going on below ground, and can we use remotely sensed spectroscopic imagery to predict what's going on below ground. In both cases, we find that diversity is a driver of productivity. But in these different systems, there are different drivers to the below ground processes. In the nutrient poor sandy soils of Minnesota, it's actually the productivity that's driving everything below ground. But in Nebraska, there's very high productivity, and it's the composition, it's the quality of the inputs to soil, it's the chemical composition, the stoichiometry of the vegetation, which species are there, and the functional group types or the phylogenetic functional types of those species that matter for below ground processes. So we have different models for predicting what's going on below ground based on what we can see from above. And so we've used spectroscopic imagery in both of these systems and found that in the Minnesota experiment, it's really all about the productivity that is driving microbial biomass, soil carbon, net nitrogen mineralization rate, soil respiration, enzyme activity. But when we are at the much more productive and nutrient-rich site in Nebraska, it's the quality of inputs. The carbon is saturated, biomass is it's complete, very high biomass in the system. So here it's really the stoichiometry. It's the nitrogen that we can remotely sense in the vegetation. It's the cellulose we can remotely sense in the vegetation, or it's the composition, it's the proportion of C4 grasses that are driving net nitrogen mineralization rates, microbial biomass, soil carbon, soil respiration, and enzyme activity. So that's just an example of how, even in the same biome, different ecosystems have different drivers of the processes that we are trying to get at. There is potential to remotely sense the planet and try to understand what's going on below ground. Factors like pH are driving bacterial composition. But we are really going to have to do the hard work to understand each of the ecosystems. So biologists have a lot of work to do. So the second point I'll make is that the Tree of Life is a really useful organizing framework. It's actually a very elegant framework because of the way that life is organized, with individuals that are nested within populations, and populations nested within species, species nested within clades of larger and larger size. That gives us information about the shared ancestry, the shared genomic information that close relatives have with each other, so that close relatives share most of their genome, while distant relatives will be very different functionally and in ways that we often can't see or measure very well. Spectral information also turns out to be tightly coupled to the Tree of Life. And our eyes see in the visible region, so we're used to looking at, let's say, plants, because I'm working on plants. When we look at plants, often we don't necessarily know how closely related they are just based on visual signals because of convergent evolution. But when we go into the shortwave infrared and places our eye can't see, often what we see across the Tree of Life, so this is a spectral signature for different major groups of plants, and this is where in the spectral region we have phylogenetic signal where close relatives are very similar to each other. So for example, the gymnosperms have very high signal in regions of the spectrum that our eye can't see. But it differs for the different major groups. But in all cases, there's very strong phylogenetic signal in spectral signatures, meaning that the Tree of Life is going to help us for monitoring the composition of taxa, maybe not to species, but to different lineages. And here's an example. So that was for leaves. The previous slide I showed you, that was people putting their data together for leaves of many, many different plant species. When we do it now for tree canopies, so pixels that are remotely sensed in neon across 21 neon sites for 101 tree species, again, we see this pattern that these red dots show where there's significant phylogenetic signal across the spectrum. And so you see for all of these tree species, each of these is a mean spectrum for a species, and again, we see significant phylogenetic signal for tree canopies. So this is going to help us for remote sensing and trying to understand which lineages we have on the ground. And these are basically Mantel tests where we regress the spectral distance versus the phylogenetic distance at the leaf level here, but also in the canopy level, in a forest diversity experiment, and then across all the neon sites. And in general, with increasing phylogenetic distance, you get increasing spectral distance. And you also get greater complementarity in species with increasing phylogenetic distance. So the spectral information is not only coupled with the phylogenetic information, but it also reveals important ecological information about how biodiversity contributes to ecosystem function. So for example, typical way that we look at biodiversity ecosystem function relationships in tree diversity experiments, like in Tree DivNet around the world, and this is the Forest and Biodiversity Experiment, Cedar Creek Ecosystem Science Reserve. If you look at species richness on the x-axis, so just counting the number of species, it predicts net biodiversity effects or over-yielding effects. The amount that different species growing together gives more productivity than you would expect based on those same species growing alone. So it's this over-yielding effect is we see all over the world in all kinds of organisms. It increases with species richness. And those different colors are just changes through time. You get increasing net biodiversity effects the longer the experiment is in place. But if you look at the explanatory importance of species richness, it isn't as high as when we use phylogenetic diversity, where we actually take the phylogenetic information into account. We get greater explanatory power with phylogenetic information. And especially when we get to, we see this very clearly when we look at two species mixtures, where you're growing two different species together. There's no explanatory power there in terms of net biodiversity effects. But when we, for those same mixtures, use phylogenetic information, we explain almost half the variation in net biodiversity effects. And that's because congeners really are basically acting like monocultures. There's really no over-yielding in close relatives, but non-congeners, the distant relatives, are the ones that are responsible for these net biodiversity effects. So using spectral signatures to understand how different species are and how phylogenetically different they are will help us understand these biodiversity effects across natural systems. And we see that mean spectral distances between species does predict their net biodiversity effects. So that's one example of how using the tree of life is informative. We can also use it for mapping species and lineages. And here's an example. This postdoc, Antonio Guzman, who's a very gifted scientist, and our NASA-funded project, is using the US forest inventory analysis, so plots that are across the entire US. We're working in the eastern temperate deciduous forests and using phylogenetic information, basically creating a phylogenetic distance matrix to ordinate all of those plots. We're then using hyperspectral observation. So in this case from DESIS, because that's what's available right now. So this is the German satellite, hyperspectral satellite that's 400 to 1,000 nanometers or 400, 900 nanometers. They're 30-meter pixels, so we're not going to get to individual crowns, but we will get communities. And this is what the spectra look like, and these are wavelet transformations of those spectra. And you can see the places where the DESIS data intersect with the FIA plot. So these are the data, the red are the areas that we have to work with. And this is an ordination, it's an MDS ordination that shows all of those plots in ordination space. So these are all the forestry plots. They're closer together if they're phylogenetically similar, and those red dots are the species, so it can back estimate where the species are in ordination space. And it can also, if we map angiosperms and gymnosperms on there, we see interesting patterns. This is the ectomycorrhizal and vesicular arbuscular mycorrhizal patterns that are very tightly linked to phylogenetic information. So this is the way the ordination was conducted. And then the idea is to use the spectral information to predict the location of plots on those axes, on those phylogenetic ordination axes. And these are the three axes, and it actually, these models work pretty well given all of this data. We get nice models for predicting those axes. And so we can then use those predictions to map those ordination axes. Here's an RGB image. Then you see the phylogenetic beta diversity based on the ordinations. And we also get the uncertainties from that. And then we can back predict approximate locations of species occurrences. So the light values are those where the species are actually occurring. A bit hard to see, but you can see the light values where those species are most likely to be occurring. And finally, the last reason that I'll give you for why the Tree of Life is an important organizing framework is that pests and pathogens often tend to be host-specific, and if we can identify the host, the lineages that those pests are adapted to, we can help detect disease. And that aids with forest management, for example. This is also really important for invasive species detection and management. And this is the example that we've been working on for the last 10 years or so. So oaks are the dominant tree lineage in the United States and Mexico. It's in eastern United States, it's about a third of the total biomass. They're also the most diverse woody group in North America. And they're being hit really hard by oak wilt disease. It's a fungal pathogen that forms these fungal pockets that you can see due to root grafting between neighboring trees, and the fungus spreads from one tree to the next. It's also spread over land by sap-sucking beetles that carry fungal spores from one tree to distant locations. So being able to find where a new infection site has occurred is really important for forest managers who are trying to stop the spread of this disease. And they actually do have effective management strategies if we can find where the infection pockets are. And this is just a valuation study that we did showing the importance of the oaks and the pines, which are also being hit by various pests and pathogens. Just in the United States, using FIA data, we were able to calculate annual value of these trees just for their climate regulation, air quality, and the wood products they provide. But even within the oaks, different phylogenetic lineages differ in disease susceptibility. So being able to distinguish the red oak group, for example, from the white oak group is really important because the red oaks will die within a year or two. They have a vascular system that wider vessels, their immune system doesn't stop the spread of the fungus very efficiently, and so these trees die very quickly. Whereas the white oaks are able to, they have narrower vessels, their immune system is more efficient at forming tyloses that block the movement of the fungus in the vessels, and they're able to sequester the disease, and they can live for decades, even with the pathogen. So it's really important to be able to distinguish these lineages. And so we have worked at a variety of different scales using different approaches, but one is using airborne spectroscopy to get imagery across forest systems for all of the forest species. And then we use classification methods for detecting species. This is not a complicated forest, 12 to 14 species. They're readily detected with high accuracy, so we can detect all of these species. And then that gives us the ability to pull out the red oak lineage. We can pull out all of the red oaks from the airborne maps. And then once we have those, we can take this phylogenetic approach where first we pull out the oaks, then we distinguish the red and the white oaks, and then we can differentiate healthy versus infected red oak trees. And when we do it that way, we get a 71% accuracy, which is much higher than about 50% accuracy when we don't use the phylogenetic information first. And as I mentioned before, if we can detect the disease quickly, then we can use these rapid response methods like chainsaw girdling and herbicide treatment. Once a pocket forms, then we have to use vibratory plows to sever root systems. And if we don't catch it in time, a whole forest can succumb to the disease, and then it's really, really hard to deal with it because it becomes a source of spores. So it's really important to get on this and keep on it, and that's what the U.S. Forest Service is doing, that's what the National Park Service is doing in the United States, and local departments of natural resources. we can also scale up physiological systems across these different biological scales, including to regional scales, which is where satellite information comes into play. And so, again, this is Antonio Guzman's paper just came out, and here he's using Sentinel-2 Landsat-8 imagery to develop a workflow to provide analysis-ready data that can be used for this kind of disease mapping. And the approach that he used was to take a phenological approach using the chlorophyll carotenoid index, which responds a lot to stress signals in the plants when they are hit with a wilting disease like oak wilt. And so you can see through time, these are just pixels through time, in a healthy tree that became sick. And you can see there's a change in the phenological shape of this chlorophyll carotenoid index from Sentinel-2 data, and then the dead signal. So he used the phenological information from this satellite data and developed these accurate maps to show us where the symptomatic trees are and dead and healthy trees are. And so now we're working with the National Park Service and the U.S. Forest Service to help provide them maps on the ground so their people can go out and find these sick trees more efficiently. So now we're at the point where all of this is exciting and great advances, but if you've seen Oppenheimer, you realize that even the best of intentions, it's really complicated if you don't have an ethical framework in place at the time you're working through all these advances, because all of these capabilities can be used for other things too. And when we're thinking about trying to save our living planet, it's not just about technology and technological advances. And I've been involved in studies doing valuation of ecosystem services, and those are really important to have a seat at the table in economic conversations. But we also need an ethical framework and really need to pose the question, what right do we have to destroy nature, going back to Chief Seattle at the beginning. And these legal ideas that have been in place for a while of, should trees have standing? Nature actually can't talk, so we have to be the witnesses and speak for the trees. And I think a lot of us have been really moved by Robin Wall Kimmerer's Braiding Sweetgrass book, where she talks about reciprocity and that science alone isn't enough. And this ethic of reciprocity is, I think, where Geobon is going. Like, we're not just monitoring, we're also trying to give back to the planet and manage, restore, rehabilitate our land. And that's what the Global Biodiversity Framework is asking us to do. And here's an example. In Kenya, I was there some years ago, and you see that all these large mammals persist because people do all the work to manage them and maintain them and give them make sure they have migration corridors. There's a huge amount of effort that goes into saving these species and making sure their populations are healthy. We do this for butterflies too, the monarchs. There's a huge effort that has gone into making sure the winter breeding grounds for the monarchs are intact. And this is a picture from some time ago with my kids. Like, we love these creatures, so we do the work to take care of them. But there's so many others that we are not doing the work for. It's going to be a huge amount of work to take care of all these species. But we've got to do this work. I mean, love it or lose it. We're not going to keep our species if we don't do the work to take care of them. Here's just one example of work that I've been involved in. So a long time ago with colleagues in Mexico, I did the genetic work for this endangered species, Quercus brandegii, in southern Baja California. And this is on the red list, and we found that it was a relict species from a much broader ancestral species from Quercus fusiformis, its sister taxon, but that it was about to perish because it's in this ephemeral riverbed in the middle of the desert, and in the 5 million years where it's separated from the other species, it hasn't been able to evolve to get out of the desert. It's still stuck in the bottom of these ephemeral rivers. And so without help, the species will go extinct with ongoing climate change. And so these folks, these colleagues, this was initiated through the Morton Arboretum, and this slide is from Sylvia Alvarez-Clare. She and other folks from the Morton have been working in close coordination with folks in Mexico, our colleagues down there, and they've started this whole program for saving this tree, doing the research, propagating the seeds, reintroducing them, restoring them, doing ex situ conservation, and doing the outreach and capacity building, so that now people are actually really caring about this tree and taking care of it. This is one example of our millions and millions of species around the world, but this is exactly the kind of thing that we need to spend these decades doing. So I'll stop there and just remind you the main points, which are Earth observations are this amazing advance in our capability, but we have to do the work to integrate those signals with biological understanding and also socioeconomic understanding for effective monitoring and management. Then I'm trying to make the case that the Tree of Life is a natural organizing framework for monitoring and managing life. I'm glad to talk more about that later. And then finally, technology without ethics and reciprocity with our living planet is not going to solve these problems that we're facing. Thank you. Oh, I'm happy to take questions. Hi, Sean.
Thanks so much for the talk. My question is about the ethical framework, which is a big passion of mine and the others at the Morton Arboretum. Do you have suggestions for those of us who are mostly researchers, how to better integrate conservation action and this change of ethics in our research projects? Sorry, it's kind of a big question, but how can we reorient our research-focused projects to include more of this ethical responsibility and working more on the ground?
Yeah, that is a huge question, and I think it's the subject of a lot of conversations that are going on here in Geobon. I mean, it's the reason that there's been so much emphasis in IPBES and the Global Biodiversity Framework on indigenous and local knowledge. I mean, it's not just the local understanding of the biodiversity, it's also the way that local people, indigenous people have, you know, when you actually have a worldview that you're living as part of nature, that you are part of nature and that you're not separate from it, you treat it differently. And in fact, they wouldn't use the pronoun it, you know. And so your question of how to do that, I mean, it's a very slow process, right? It's not like you just come in and change things. It actually involves listening to people and spending time together and trying to increase understanding. And we do have projects with the Menominee Nation for monitoring oak wilt. And the way that's working is it's really a student who is leading that project on their land, and we help with the technological know-how, like training, providing training, and also providing them maps. That's what they want. But it's a lot of spending time with folks in the same room and listening to what they're trying to accomplish, and really trying to figure out what they're after. So it really goes back to this co-generation of knowledge and taking the time to listen and understand where people are coming from. I think there are other people in this room who have done a lot of work and thought a lot about this who should probably be answering your question. So I'm glad for other people to raise their hand on this issue.
I will follow up with a question. Not on this topic, though, as important as it is. That was a tour de force. Thank you so much for that presentation. And I'm learning so much from just the progress that you and your team are making. You didn't speak so much about the fundamental changes in biological time. I think part of the issue is that humanity is driving things at a certain pace. We're having trouble keeping up with the impacts and consequences of that pace. And then there's life on Earth and the rhythms of life on Earth. And so how does spectral information give us something about that change there?
Okay, I think what you're saying, Andy, is that the rate of change is faster than it has ever been. I mean, if you've just lived a few decades, you've experienced the rate of change. It's dizzying, right? Like you just figure out one technology and then we have the next one and the next one. I mean, we grew up without cell phones, right? We grew up without the internet, and like here we are. So there's this rate of change in the way we live based on technology, and then there's the rate of change in the planet. And I mean, it is shocking to see the rate of extinction, the rate of ecosystem change, the rate of land use change. I mean, it's really, really hard to see that. But now we will have the capability, if we do the work, to have near real-time ability to see the whole planet at once and how it's changing. And that is rather mind-boggling when you think about where we were when we were kids. Like, what? That you could see the whole planet and see how it was changing and really understand in depth the various kinds of changes that are going on all over the world. So that gives us, I mean, I think just even the imagery of the whole planet and understanding how it's changing and how it's fragile gives us a different conceptual framework. And it does give us an ability to manage things much more efficiently. I mean, this is certainly the case with the disease detection that I'm involved in, where we do the detection and then people are on the ground looking for those trees and are dealing with them that season. So that's just one example, but we need millions and millions and millions, billions of examples like this where we're responding in real time to the signals that we're getting so quickly from space. Other questions?
Thanks. Thanks, Jeannine. I'm wondering, because remote sensing can be useful in terms of prioritizing conservation because of its relationship to lineage, do you have recommendations for space agencies in terms of what they should be doing to facilitate that?
Okay, Gary, I can't see where you are, but I recognize your voice. There you are. Okay, we're going to work on that, Gary. I'm going to try to explain that at the AGU meeting, unpack it a little more. And so actually, we have a paper coming out for Frontiers in Ecology and Environment. It's called No Branch Left Behind. And it's basically making the case that if we want to preserve the innovations, the genomic innovations that life has generated over the 2 billion years of evolution of life on Earth, we really need to make sure we keep every branch. And I'm not just saying phylogenetic diversity. I'm not talking about a metric. I'm talking about an accounting framework. So really like a list of all the branches of the tree of life. How do you come up with a branch when you have a fractal system, a hierarchical system like that? So the paper presents an approach to explaining what the branches are, and there will be tools that we generate that give you all the branches. And then let's say that you're a manager for the national park system. You have your park that you're responsible for managing, and then you have the set of lineages, the set of branches that are in your park that you're responsible for maintaining. It's basically an accounting framework. And the reason why that's relevant for space agencies is that it's easier to detect a lineage than it is a species. So there's a practical aspect to it. Like, it's hard to differentiate every single red oak, but we can differentiate the red oaks from the white oaks. So, for example, and they're like so many examples like that, where you go to the tropics, it becomes even more important, because often biologists don't even know a lot of these species haven't even been described. You go out there, we don't know what they are, but we might know which major lineage they're in. And so at least having that information, having this hierarchical tree of life as an accounting framework, what do we have now? How is it changing? You know, at least getting it to lineage, which we can do from spectral signals, has advantages from a practical perspective. So there's two sides of this, practical and in terms of conserving the genomic innovations that have been generated on planet Earth. I saw a hand over here. Yeah, go ahead.
Well, I was recently told that, well, I work for ecosystem accounting, and recently I was told that, okay, but what do I do with this number? And it was an economic valuation, and to be honest, I was doing my work. So what is your view then in relating all of this information that we're gathering for the ecosystems and to translate this into well-being and health benefits? For example, air filtration, water purification, but also link it more to morbidity and these things. Thank you.
Yeah, it's a good question. I'll just say I'm a biologist, but I have collaborated a lot with the Natural Capital Project, particularly Steve Pulaski, who's also at the University of Minnesota. And we've taught together a sustainability science course for many years. And I was convinced for many years that doing the valuation work is the answer. Having an anthropocentric perspective is the way that you reach people besides biologists. I think a tiny fraction of the Earth's population actually thinks about biodiversity the way that biologists do. So I learned to just, okay, let's take an anthropocentric perspective and do the valuation, because it gets you a seat at the table, particularly in the United States. It's so money-focused. Putting things in terms of dollars is an important way to compare costs and benefits and to look at trade-offs. And I think that is true, but I prefer, the Natural Capital Project actually is really clear that they don't just do that. They also are putting things in terms of units of water, gallons of water that are conserved, the set of species that are conserved, the energy that is saved by having shade, for example. All of these kinds of things, you don't have to put it in dollar units, you can put it in terms of the benefits to people that actually matter. And the whole nature's contributions to people was an attempt to sort of get away from putting dollars on things, especially because with disparities in wealth, a dollar doesn't mean the same thing to different people. So dollars can be very problematic, and especially when, like, you realize that the oil sitting underneath the Arctic is more valuable than the services that you calculate for it. It can be complicated to use dollars. But so I think what people really care about should be the y-axis on the things we're predicting. And I think there's a huge value to doing that. But I also think we still need an ethical framework. I mean, I feel like maybe with COVID we have reoriented a bit. We do feel like we're part of nature and we're not something separate from it. But I think that's where we need to get, is that we actually have to give back to this planet, and because we're part of it.
Okay, thank you. Maybe we can get one last question. Okay. Sorry.
Hi, Jeannine. Thank you for that talk. Very, very inspiring. I wonder how you take these different studies that are done by a student or postdoc or a small group to something operational that I think maybe that goes with Gary's question. What is the product that, or the few, maybe 2 or 3 products that we should think about in asking these agencies to produce to help us move along?
Yeah, I think that's you, Frank, right? It's hard to see with the light, but okay. So NASA wants products from these. So that's what we're working towards. So we're working towards figuring out what we can generate when SBG is launched and that data becomes available. We were working with DESIS now because that's what we have. So I do think real-time updatable maps of composition using a phylogenetic framework will be really important as one product. The trait maps that are going to be possible to generate are another. And we're just one group, but those are things that our group is really focused on. And then I think there'll be so many others that fit into the EBV framework. And I know that the Europeans are thinking about this quite rigorously and have already got things sorted out in a way that might work now. But I think we're working on that. And I think it's going to be ongoing discussions, figuring this out, doing the next steps after this initial work that says, this actually works. Hey, wait, we can do this. We can do this really well. We have all this training data, and we are getting good validation results from this approach. We can scale this up. So that's not a very satisfying answer, probably, to what you're thinking. But maybe you want to say more about what you're thinking?
This discussion during the coffee break. I'm so sorry to cut you, but we are running a bit late behind the schedule.
Okay.
Thank you so much,
Yeah.
Jeannine, for this wonderful talk.